Still we are in the beginning phase of learning microanatomy. These are the basic items that is the foundation for learning the histology. So we have an understanding of this particular element of the tissue, cells, etc., etc. So I am going through this understanding of these tissues, organ, etc., etc., and we will be building through. Now in 10 plus 2 we have learned about these particular cells, tissues, organs, etc., etc. So we have an understanding how these particular things are built. Then we call them as cells, which together forms tissues, and then its organs is formed, which has a particular form of actions. So this is the basic orientation in simple. Now our point of interest is this one is tissues. So we have plenty different kinds of tissues in our body. So we will be catching up one of these particular tissues today and break it down to understand how this particular tissues is working here. Now in the tissues which we have here, we have several different types of tissues. We have the glandular tissues, we have these particular connective tissues, we have this muscular tissue, nervous tissue, etc., etc. We know this particular breakdown of the different forms of the tissues. Now in histology today we are going to talk about the tissues. We catch up this tissue, we call them as the glandular tissues or the glands, which are kind of like a prime thing whenever you are seeing any organ or seeing any histological structures. Now having said that, so the definition of tissue, you have any understanding on definition of tissue? We know about it. So similar kind of cells which is placed in a common place, which is having some kind of common goal or common actions or kind of like a similar kind of actions which is performing to help you out any physiological functions of the body. If you just like make it very simple in terms of like working as a definitions of the tissue or what exactly these particular things are. So if these particular cells are together and working a similar kind of actions which is producing in here, in this today's class we are going to talk about the glandular tissue. The functions of the glandular tissue is produce a secretion. So this secretions which is coming from this particular glandular tissue, which is a point of concern. Now how these particular tissues or glands or glandular tissues will be classified. So we have to know what are the different types of tissues in other short. If you just like do not want with this particular complex word to hit our head like classification, etc., etc. So we should be knowing what are the different types of tissues or glandular tissues exist in our body. Now in a basic way to differentiate this glandular tissue is that one having a kind of tube or one do not have a kind of tube. Now why this particular tube matters to us? That as we have seen, this particular glandular tissue is doing or producing this thing which is known as secretion. Now this particular secretion which is been produced from this gland need to be taken out or needs to be getting in a wider dimension, need to be spread out. Now if this particular secretion is coming through a tunnel system which is known as the tubes, we call this particular tube which is linked to it as a duct. So if it is having a duct, we call them as exocrine, and if you do not have a duct, we call them as endocrine, right? Am I saying the reverse? I have a tendency of saying the reverse thing. Don't worry. So if you have a duct and no duct, so the no duct thing we call them as exocrine. We call them as endocrine. So no duct is there. Only the blood circulation flows through and takes the secretion from it. If you have a duct, we call them as exocrine glands. That means you have a tissue which is producing the juice and it is thrown out through a sewerage system, drainage, drain দিয়ে চলে যাচ্ছে, গিয়ে বাইরে চলে আসছে to a place and then to a smaller place, bigger place, etc., etc. Say for example stomach. In a stomach we have this particular gland coming from the gastric glands. Stomach we call them as gastric thing. So gastric gland producing the secretion through a tunnel, it is coming out, we call them as gastric pit. The gastric pit empties into gastric canal and getting inside the cavity of the stomach. So it is coming through. So no blood circulation is required for this. But if we need to produce some secretion from the thyroid gland, likewise we do not have a duct system which is the secretion is coming through it. So we have a blood channel system, capillaries, which is flowing through, is kind of like a Ganges or rivers flowing through. Whatever the extract from this particular glands, which is endocrine, is there, it is liberated or emptied into the blood system, and the blood flows through the different parts of the body and runs through different cells and different parts of the organ. So it is flowing through rivers, which is the endocrine system. Now largely for this class we will be focus our discussion on the exocrine glands, little bit of the endocrine glands as well we will do. We will do a dedicated discussion of endocrine, of individual endocrine gland whenever it is coming, like thyroid gland, like parathyroid gland, like adrenal gland, etc., etc., pituitary. So we will be focusing this discussion on the exocrine part. Now to break this particular thing down, to make it a simple about this particular gland. So what this particular gland, which is exocrine variety, is composed of. Now what is the basic thing about this particular glands they are doing? They are producing secretions. That means they have a factory to produce the secretions. So this is the factory of producing secretions, and once this factory has produced this particular secretion, it has to be transported out. You need a tube or pipeline to take these things out. We have a thing which is there, right? So we have a component of this and we have a B component on this particular exocrine gland. Agreed? Right. Now this A component we call them as secretory unit. We call them as secretory unit or else the factory. The secretory unit in our histological words, secretory unit in our histology words, we call them as AC9. We call them as AC9. Make sense? Comes to have a single duct or many ducts, we can classify the names. If you have a single duct, one only, we call them as simple. If you have many ducts, we call them as compound. What do you call them as? Simple or compound, depending on counting the duct numbers. Now how this particular thing, duct number is present here? It may have a single duct which may have a branching. So initially it was single, later on it has a branching, and then this particular thing, branch thing, is penetrating this particular secretory unit and became a compound thing. So this particular simple duct or single duct, we call them as simple kind of gland, and if you have many ducts which is known as branched, we call them as compound gland. Make sense? Simple or compound, right? Now apart from this particular duct, we go down to the secretory unit. We call them as AC9. Now this particular AC9, how this particular AC9 look like, we can classify them. So this particular factory house may look like a branch like thing, grapes. They may look like grapes, or they may look like tube, or they may look like tube and branch then. So if this particular AC9 secretory unit looks like tube or looks like this particular grapes or this particular both things, we call them as alveolar pattern or tubular pattern or tubulo-alveolar. Mix of things. No complex things, just use the thing, use your common sense, common knowledge. Tube টাকে কেমন দেখতে? Tubular kind of gland. If it is a branch, grape এর মতো দেখতে হয়, alveolar pattern. If it is mixed of things, partly tubular, partly kind of alveolar or grapes like, we make it like complex thing, which is tubulo-alveolar. Now if they are fitted with one duct, we can call them a simple alveolar. If it is single duct which is tube fitted with, we call them a simple tubular kind of thing. If they have this particular many ducts, we call them as compound alveolar, compound tubular, alveolar. এভাবে classify করব। These are the basic form of classification. It has nothing to do with us because we are going to be doctors, right? We are going to earn money. We are going to treat patients. We are not going to dig deep into what kind of tissues or glands, etc., etc. But there is one form of doctors is there. We call them as pathologists. They have to dig deep into it. They have to know a lot detail about it. So what kind of gland is there, what transformations of gland is there, what kind of transformation gland or any bad transformation is there or good transformation, they have to know it. Now that particular science you cannot study unless you know the basic thing. So to study this pathological division, this is a foundation thing. This is not very complex thing unless you make it very complex. So simple form, just try to use a logic. How does this thing look like? How this classification is based? How this particular things is coming? We can classify this. Fine. Now other form of knowing this, what are the different types? We call them as classification, or what are the different types. Now we take this particular secretion as it. So the secretions or secretory part which is coming. Now if this particular secretion has a particular consistency, we call this as a consistency of the secretion. If it is a watery in consistency, if the secretion is watery in consistency, we call them as serous. If it is sticky, we call them as mucoid. Understand? So if we have the secretion which is coming from the secretory unit is watery in nature, we call this as serous. And if it is sticky, we call them as mucoid. Make sense? So nature of this fluid which is coming through, we call them as serous acini or mucus acini. That means acini is producing watery fluid or watery secretion. We call them as serous acini. The acini is producing mucoid or sticky element. We call them as mucoid acini. And that particular way we can classify as well. So no big deal about it. So just don't get complexed or perplexed with these particular names, whatever been used. Use your head what this particular element is trying to say us. It is basically giving a clue to identify this nature of the glands or nature of the secretions of the gland. They are giving the answers to it, right? So by knowing this particular name, what does it mean? It helps us to basically to decode the functions or the element what this particular thing is doing. Okay. Similar to that, we have another way of classification, how these particular secretions are coming. So this factory are producing the fluids. Now how these particular factories are producing the fluid? So we have three types of classification which is based on this particular nature of mode of secretion is there. But that means modus operandi. Say factory is producing, a part of factory is blown out. We call this kind of factory which is secretory part which is kind of blown out a particular variety of the glandular tissue. If its entire factory is blown out, we call them as another type of variety. So we are familiar with these terms. We call them as holocrine, apocrine, merocrine kind of secretions. So if the secretions or these particular cells which is coming from it partly blown out, we give it a name. If this particular secretion coming from this particular slice is completely getting out, we give another name. Holocrine, that means entire gland or entire cells is kind of destroyed when the secretion is done. If it is merocrine, which is having a different pattern, and another pattern is, another type of secretion pattern is apocrine variety. So if upper half of this particular gland is blown out, we call them as apocrine variety. So there are different types of glands, serous gland, sebaceous gland, zoid gland, which is basically of different types. So these are the basic hallmark or architecture how we classify the glands. And this particular classification, based on this, we identify the tissues and build on this particular concept of it. Now, in histology, whenever we are seeing these things, we are seeing two-dimensional element. That means a tissue is cut on length and breadth, whatever it is, two elements are taken, stained and seeing it. In our body, we are not two-dimensional element, we are three-dimensional element. Whatever the content is there, where from the tissues are there, it has three dimension. It is a 3D structure. But what you are seeing here is a 2D structure. So, whenever you are seeing this particular microscope, through microscope, if you are going through or cutting through glands, some of this particular secretory element may look like circular or some of them may look like tubular. Now you say that there is a mix of things, there are circular kind of like elements is there and tubular element is there. No, it's not like that. So some of the glands are in a tubular fashion, which is cut through, is showing a tubular pattern. And the same kind of gland has a different orientation, has a different plane. So that particular plane is maybe just like bisecting this particular sections. So that's why that some of the section may appear like this, tubes like this, and some of them are circular like this. So you are seeing this particular glands which is like this, and some of them are this. So these are not the two different elements which you are seeing here. These are the same thing. Some of the sections are like this and some of the sections are like this. So some of them are looking horizontally and some of them in a cross section. So that's why they are taking two different shapes, but basically they are of same time. So it is happening because you are seeing that structure in a 2 dimension. It is a 2D structure. So if you just like mix in and see it from a different direction, it is a 3-dimensional thing, which will be always also looking like the same. Alright, so I am projecting some of these particular slides based on these particular glands. How does it look like? So it will clarify and clear your mind. If you see them, then we can see this on the microscope. This is what we are saying. Now this is the formations of these particular glands. Now there are 2 types of glands we mentioned. Now there is a one type of gland, we call them as endocrine, other form is exocrine. The tissues from where these particular glands are coming, we call them as epithelium. So there are 3 basic types of tissue we have: ectoderm, endoderm and mesoderm. Mesoderm means muscles and etc. etc. Endoderm means this particular GI thing which is inside your mouth and everything. And ectoderm is the outside thing which is the skin part. So these particular tissues or the glands, be it endocrine, be it exocrine, develops from the surface ectoderm, the outermost layer of the body. Now there are difference in these formations of these particular endocrine and exocrine kind of glands. Now this is how this particular things are formed. Say this is a surface ectoderm, a tissue is invaginated, that means penetrating, and then this particular ducts is formed. So initially it is having a duct like this, and gradually this ducts is branching out and becomes a compound kind of gland. So we call them as multi-branch. This means compounding. That means it is growing and making some further branches. So this is how it looks like. This is the ducts which is going on, and this is the secretory unit, the factory house. You see that? This factory house looks like grapes, bunch of grapes. So we call this as acini, which is secretory unit. Alveolar pattern, we call them as alveolar acini, these elements. Now if you see these particular cells, these are the pyramid kind of cells. You see that? Triangular cells are there. On top of that we have a cell resting on it. This is a kind of flat cells. You see the difference of the cells? Some of them are triangular, which is highly stained, where I am putting the cursor on this triangle. Can you find it? And top of here you see that there is a tiny tissue which is flat kind of cells which is on top. So this particular acini or the secretory unit has a secretory cells which is this one, triangular shape, and then we have a flat shape on top. The flat shell on top we call them as myoepithelial cell. That means these are the muscle cells on the top of it. This myoepithelial cells contracts. They have an filament which is known as actin and myosin kind of things, which is contractile tissues. These contractile tissues are present within the cytoplasm of these particular cells. Now they contract. What is the functions of these myoepithelial cells? Once this contract, they are muscle thing. They squeeze these things. That means we are squeezing an orange or squeezing a lemon. So once you squeeze this, the juices come out, right? The same thing is myoepithelial cells are doing. They are muscle cell squeezing these epithelial cells, which is producing the secretion. Once they squeeze, the secretion are poured into this particular duct. Once this particular duct is kind of getting the secretion, it is liberated out through this duct system. And over here as well we have this particular ducts which is having a contractile nature. So this particular secretion initially occupies here and then moves out with the pumping of the ducts. So this particular acini or secretory unit has a secretory kind of cells and as well as a squeezing kind of cells. What's the name of the squeezy cell? Myoepithelial cells. That means muscle of epithelium. Epithelium is a kind of tissue or cells which is lining on here. So this muscle epithelium cells over there, they look like half or flat, half demules or half cells or flat cells. They have different kind of cells, but forget about this. So this is a basic thing how this particular thing which is grown or formed here is an exocrine gland with a duct. Now similar thing with this development of this endocrine. You see that they are sourcing from the same epithelium. Now they are digging deep or invaginated, and once they find a blood vessels or capillary, they break free from the surface epithelium. See that they have a connections through here coming out here, and they break free these particular cells, and eventually they conglomerate around that particular capillaries and settle there. Now they dig into the capillaries and this particular glandular cells which is growing around the capillaries. So these particular cells also have this particular cluster element, myoepithelial element also present. They have the secretory capacity, but they don't have any duct yet. What they have instead, they have the capillaries fitted in it. This particular capillaries, they can empty the secretion into. Once this capillary gets this secretion, they runs through the body system, through this circulatory system, reaches to different parts of the body through the pumping of the heart. Clear? So this is the modus operandi and the functional thing, how these things are developed. So instead, this particular thing, if this thing was not detached from this could have formed a duct. Now if this thing was persisted, then it is transformed into duct, then this element could have been a ductal tissue. That means they could have then a exocrine tissue or exocrine glands, as because they are signaled or programmed to form a detachment like this. So they will be forming an endocrine cluster and they are placed accordingly. Get it? So this is how these two elements are produced, but you do not have to remember things. This is for your understanding how these things, to clarify it, then it will be coming with the logic. Now this classification based what you call over here. So if you have a cells, how many cells that particular element or tissues are forming. If you have a multi-tissue, that means multicellular tissues. If you have a single gland, one gland tissue, we call them as unicellular glands. Exocrine, endocrine, and the manner of secretion, how these particular things are coming out, how the factor is going out, completely blown out, partly blown out, or not blown out at all. So entire cells is destroyed, part of upper cell is destroyed based on these things. So talking about the exocrine tissues, we talked about the glands. So this is the duct, you see that simple form, and this secretory portion. This is the basic architecture. Now if this particular secretory portions is branching out like this, we call them as acini, glandular, simple branch. One duct like this, then we have this. We do not have existence of this kind of gland, like simple duct and a single acini, simple alveolar acini. We don't have this. Alveolar cells or alveolar pattern, which is grape-like thing, not existing. We have a different type. So this is how we are classifying it. Simple tubular coil, simple branch tubular. So this has a branching which is going into it. That's how we are classifying it. So simple tubular glands. If this particular glandular secretory portion or acini looks like tube, we call them as this. Now we call them as compound, which has a branching, complex branching like this. So compound branching is there depending on how these things is grape-like or tube-like or mix of these things. So it is a tube and as well as this is a grape. So we call them as tubulo-alveolar pattern. Get it? Make sense? How we are classifying it? This is how. We do not have to remember all these things whenever identifying this tissue. But you should be knowing they are existing. We may see or may not see, doesn't matter. If you do not see, that doesn't mean that do not exist. So you are not seeing in Japan nearby, that doesn't mean Japan is not existing, right? So this is how these things we are classifying. So this is how when you stain it properly looks like. It's a branch of acini which is lying this. This is a branch or duct system which is present, and this is a secretory unit. This unit is clustered together and producing the secretions, right? This is how this particular in real super microscopic structure it looks like. You see that this is a tube which is seeing here, and this central part it pierces through is the duct. So all the secretion from here is coming through and getting into it. Make sense? How these tubes are glandular pattern looks like this. So this is a cluster. As I am saying, this is kind of like a section which is taken over here. Now you see that this same tubes looks like circular elements. Now you say that this is branching which is look like tubes. Do not look like tube, rather it is circular. No, it is not circular, it is tubal, but the section which is taking here, it is because of the orientation of the gland, it is like this. It is a cross section which is taking. And why it is so? Because it's a three-dimensional element converted into two-dimensional thing. That's why it is looking like this. If you are seeing that in a real 3D, it would have been same branchial pattern, likewise the previous one. like this, tube like this. So this is also the same tube, is looking circular because this has a different plane of orientation. That's why some of them are looking like circle, some of them are tube. Make sense? So this is how this particular acini which is looking like. This is a duct which is present, and this pancreatic tissues which is surrounding to it, it is lying there. So same duct which you seeing that, and it is branching like this. This is the circular element, as I am saying. The central part of the circular here where I am putting the cursor is the duct. So this same duct out here, it is a longitudinal duct, and this is what you are seeing, the circular, because of the different orientation, different plane. So you have to orient yourself whenever you are seeing this element. So you have to narrow it down, why this is this and why this is that. Doesn't matter how does it look like, but this is how it is circular and why it is tubular, which is looking at the same tissue. Now you see that this is a duct and this is a glandular element. You should be able to train your mind to identify which one is the duct and which one is the factory. So this is how you have to look into the duct element you have to find, and as well as the secretory unit. Secretory unit means acini. So duct is a central cavity and is lined by a cells which is flying over here, and it is having a cavity, central cavity, which is the glandular element do not have it. So this particular glandular element is this one, and this tiny white thing is the duct unit. Duct in it, inside, penetrating to it. These are tissue spaces, septas, which is partisecting, bisecting out, separate করে রাখছে, মানে এটা আলাদা আলাদা করে রাখছে. It is a dome or districts. আলাদা আলাদা জেলা হয় না, এই জেলা ওই জেলা, boundary kind of thing. So it's kind of districting out, septas. Septas গুলো করে রেখে দেয়, ছোট ছোট করে পোর্শনে ভাগ করে দেয়. আমরাও তো আমাদের দেশটাকে ভেঙে 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 ছোট করেছি. So similar thing, we take these ideas from here. So we split it in a smaller portions and encircle it. Each of the districts is governed by district magistrate, etc., etc. So these things also have a particular blood supply, particular nerve supply, particular lymphatic drainage, and a particular modus operandi. And eventually, it's a country as a whole. So same thing. Everything, whatever we are producing or making, is taken from our body. We are not doing anything new. More we discover and more we implement how these things are governed in our system. So our system itself is guiding us to operate and do the things. So classification based on this particular chart will help you out to understand this branching pattern, etc., etc. What do you call them? A simple, tubular, coiled, branch, etc., etc. Compound, simple and compound, what is that, and how these things look like based on this. Alveolar, tubular, alveolo-tubular, these are the things. So this should not be any like a difficult to understand. Now these examples of this particular element you do not have to remember. Just know this classification pattern. These names, just don't run after these names. Like intestinal glands, simple tubular, these things, just don't try to memorize these things. As you go through, it will stick to your mind. Just like don't overdo these things. Just remember this upper classification. The example part, don't remember this. Alright? You will coming into it. How this nature of secretion is there? Serous or mucus. That means water or sticky or partly. Make sense? So everything together we can classify. Serous acini and mucus acini. Now what we should be knowing, how these things is going to look like whenever seeing on a microscope. The mucus thing, which is bright in color, serous thing is dark in color. If you go by simple logic of understanding, the serous and the mucus have a different context. The serous thing is rich in proteins. The mucus is rich in water. Serous thing is rich in proteins, and the mucus one is rich in water, jol. So that's why likewise this one is looks like icy thing, বরফের মতো দেখাচ্ছে, water thing, frozen, and this one having pyknotic. That means it is no nature of the fluid. It's not like this. Now whenever you sing, I am not talking about this how the fluid is coming. I am saying how you are looking into it. যখন দেখবে মাইক্রোস্কোপে জিনিসটা দেখতে কেমন, মানে আমাকে যেরকম দেখতে আমি সেরকম নাও হতে পারি, আমি খুব রাগী লোক, সো দেখে মনে হচ্ছে না নিরীহ লোক, যে কিছু করবে না, কিন্তু রাগী লোক. So the look is something and the function is something, right? So this secretions which is coming here is over here. The stickiness is because of the mucus which is present in here. So this is enriched in the zymos and granules. You see the red particles over here? So it's called them as secretory granules. The secretory granules are rich in proteins. Now the dye which you are using, that sticks to them. আর এইখানে এই জাইমোজম গ্রানুলস গুলো নেই অতটা, দ্যাটস ওয়াই এটা স্টিক করছে না, ইট ইজ নট স্টিকিং টু ইট। সো দ্যাটস ওয়াই ইট ইজ টেকিং হাই স্টেন অ্যান্ড দিস ইজ টেকিং লো স্টেন। মানে স্টেনিং যেটা রং দিচ্ছ যেটা গায়ে লাগছে না, আর এর গায়ে লেগে যাচ্ছে। লাগার কারণটা হচ্ছে যে কিছু একটা আছে ভেতরে যে ধরে নিচ্ছে, যার ভালো লাগছে বলছে বাবা ভালো লেগেছে, এটা রেখে দিই তো রেখে দিচ্ছে, ভালো ভেতরে থেকে যাচ্ছে, ও যে না লাগবে না, আমার কিছু লাগবে না, কিছু লাগবে না, ছেড়ে দিচ্ছে। ঠিক আছে, তার কারণ হচ্ছে যে লাইকহুড নেই, মানে ওর ভালো লাগে না তাই নিচ্ছে না। ঠিক আছে, কারোর বিরিয়ানি ভালো লাগে, কারোর সেদ্ধ ভাত ভালো লাগে, বলো। না no, it's not about this particular nature of the things. What I am saying, how does it look like? ওয়াটারি মিন্স এটা ফ্লুইডি, নেচারটা ফ্লুইডি যখন তুমি দেখছো, বাট আমরা তো ফ্লুইডটা দেখতে পাচ্ছি না, আমরা কি দেখছি? স্টেইনিং দেখছি, সেলটা কেমন দেখছি। তো আমার যখন আমি মাইক্রোস্কোপে দেখবো, তুমি এখানে দেখতে পাবে না কোথাও যে এখান থেকে মিউকাস সিক্রেশন হয়ে যাচ্ছে, এটা দেখতে পাচ্ছি কি সেলটা কিরকম। আর এটাও কি দেখছি? আমরা সেলটা দেখছি, আমরা কিন্তু সিক্রেশনসটা দেখছি না। So whenever you are seeing the microscopes, we are seeing the cells which is stained. This stained part which is present here, the secretion, nature of secretion we are not considering here. So don't get into it, amra secretion dekhchi. What we are seeing here, the nature of the cell, the cytoplasm and the content of the cytoplasm. এখান থেকে কি ফ্লুইডটা বের হচ্ছে আমরা কিন্তু দেখছি না, neither we are seeing this one. This particular mucus which has a sticky nature, which we are not appreciating here. What we are appreciating here, how these things looks like. What I am trying to say is that what exactly they look like, they are not performing this. So আমার কি করতে হবে, কি কাজ করছে সেটা দেখতে হবে না, জিনিসটা দেখতে কেমন আর দেখে চিনবো কেমন করে সেটা বুঝছি, কারণ ওটা আমি দেখতে পাচ্ছি না, লাইভ অ্যাকশন তো দেখতে পাচ্ছি না, এটা তো ফ্রোজেন বিলিয়ন ইয়ার্স আহেড। কবে স্লাইড বানানো হয়েছে? ধরো 15 বছর 20 বছর আগে, তো এই কাট সেকশন ফ্রোজেন। এটা যেটা তুমি দেখছো, ইট ইজ আ পিকচার অর সিলুয়েট টেকেন 10 years 14 years ব্যাক। কি দেখছি আমরা? সেলটা দেখছি, সেলটা কিরকম দেখতে দেখছি, ব্রাইট স্টেইন অর নো স্টেইন। এই স্টেনিং প্যাটার্ন দেখে আমরা সেলটাকে আইডেন্টিফাই করছি, বাট আমরা কি ধরনের সিক্রেশন করছে সেটা জানছি না। সেটা জানতে গেলে আমাদের অন্য কিছু করতে হবে। So what the purpose of being here in histology thing, we are identifying, we are not appreciating the action. The action part is physiological or physioanatomical, which we are not seeing here. Histology is nothing but movie picture. আমরা অ্যাকশন অর ফাংশনস দেখতে পাচ্ছি না, পাবো না। এখানে যখন আমরা অ্যানাটমি দেখছি, it's a dead cadaver, we cannot see the muscle is working, but this muscle we can see. যদি মাসেলটা কি করে বা কি অবস্থায় দেখতে হয়, তাহলে আমাকে ফিজিওলজিক্যাল এক্সামিনেশন করতে হবে, যেখানে ফিজিওলজিতে করে যে একটা মাসেল, একটা ফ্রগ লেগ, তাকে ইলেকট্রিকিউট করলো, তার মাসেলটা কন্ট্রাক্ট হলো, we can see that. Then we are seeing the muscle is contracting, but here we cannot see this muscle is contracting. যখন আমরা ডিসেকশন করব, লোকটা উঠে ঘুষি মারবে না, দেখো কিরকম মারলো ক্যাডাভারটা, it's not happening. So what we are seeing, the muscle we are demonstrating. How the muscle is working, I cannot show you there. Either you have to go to the live OT theater or you have to go to the physiology lab, fresh leg, যদি একটা ফ্রেশ মাসেল পাওয়া যায় সেটাকে ইলেকট্রিকিউট করে মাসেল কন্ট্রাকশন সে তখন দেখা যাবে, মাসেল কন্ট্রাকশন করে গ্রাফিক রিপ্রেজেন্টেশন দেখা যাবে। Understand? So what you are seeing here is the cell nature, not the action or what the producing factor is there. ওই যে বললাম মায়োএপিথেলিয়াল সেলস কন্ট্রাকশনস, ওই ডেমিনুলটাকে ওই কঠিন নাম দিয়ে দিয়েছে, বুঝছিস তো? ওটা কঠিন নামটা মনে করতে হচ্ছে না, ঠিক আছে? ও জিনিসটা হচ্ছে, the thing is that it's a muscle of the cells. কিছুটা আছে গোল, we call them as serous demilune, কিছুটা আছে হাফ, we call them as hemidemule, মানে মাসেল সেল কি মাসল সেল? মায়ো এপিথেলিয়াল সেলস। So mucous gland, this one. সেরাস গ্ল্যান্ড কোনটা হবে তাহলে? ডিপ কালার যেটা। বোঝা যাচ্ছে কেন বলছি? ডিফারেন্সটা বোঝা যাচ্ছে একই পিকচার। পার্টলি সেরাস, তাহলে এইটা কি টাইপের গ্ল্যান্ড বলবে তোমরা? যেখানে সেরাসও আছে মিউকাসও আছে, মিক্স, তাহলে মিক্স ফ্রাইড রাইস, তাই তো? গুড। এর ক্লাসিফিকেশন আছে, এর টাইপস আছে, নেচার অফ সিক্রেশনস রয়েছে, ব্লা ব্লা ব্লা, এগুলো পড়ে দেখে নেবে। সো দিস ইজ দ্য সেম থিং উই আর টকিং। সো ডিফারেন্স রয়েছে, এগুলো বইয়ে অনেক রয়েছে, আমি বলে আর সময় নষ্ট করব না, ঠিক আছে? কি ডিফারেন্স রয়েছে অফ দ্য গ্র্যানুলস, এই জাইমোজেন গ্র্যানুলস আর মিউসিনোজেন গ্র্যানুলস, দে আর পার্টিকুলারলি ডিফারেন্ট টাইপস অফ গ্র্যানুলস। এই গ্র্যানুলস গুলোর জন্য স্টেনিংটা ডিফারেন্ট হচ্ছে। এতগুলো কথা না বলে আমাদের এইটুকু মনে রাখলেই চলবে, ঠিক আছে? দে হ্যাভ এ পারপাস। ফিজিওলজিতে আর একটু পড়বে এই ফাংশনস অফ দিস পার্টিকুলার এলিমেন্টস, আর কিরকম কি দেখতে লাগছে, অ্যাসিনা ফ্যাসিনা, এগুলো জানবে, এগুলোর মধ্যে বলার কিছু নাই। বেসড অন মোড অফ সিক্রেশন, এটা আমরা ওই ক্লাস 8 থেকে বোধহয় পড়ছি নাকি? হলোক্রাইন, অ্যাপোক্রাইন, মেরোক্রাইন, কে মরলো কে বাঁচলো ওই সমস্ত। এপাকাল পোরশন উড়ে গেল, তার মানে অ্যাপোক্রাইন কাইন্ড অফ গ্ল্যান্ড, পুরো গ্ল্যান্ডটাই ধ্বংস হয়ে গেল, তাহলে কি? হলোক্রাইন। আর থেকে গেল কিছু ঢুকেছে, মরে গেল, হ্যান ত্যান, এই সমস্ত আর কি। অল দিস থিংস উই নো বেটার দ্যান মি। সো দিস আর দ্য থিংস হাউ দিস পার্টিকুলার থিংস ইজ ক্লাসিফাইড। সো অ্যান্ড এক্সাম্পল, আমাদের বোধহয় ছবিও আঁকতে হতো পরীক্ষার সময়, লেখো। এখনকার দিনে হয় কিনা জানি না, আমাদের সময় ছবি টবি আঁকতে হতো। আমার তো ছবি দেখে সবাই ভয় পেয়ে যেত, বলতো ভাই তুই ছবিটা আঁকিস না, বাদ বাকি সবকিছু করিস, হাতের লেখাটা একটু ঠিক কর। আমি তো ক্লাস 9-এও পর্যন্ত অ আ ক খ এবিসিডি লিখিয়েছি, আমার হাতের লেখা এত ভালো ছিল, বুঝতেই পারছিস আমার হাতের লেখা দেখে। সো these are the elements about holocrine gland, apocrine gland, merocrine gland, এটা নিজেরা একটু পড়ে নেবে, দেখে নেবে। This is about endocrine tissues, paracrine and autocrine kind of things. এটা কিছুই না, আমরা জানি কিছু 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 ব্যাপার। মানে কি endocrine glands, এটা এতক্ষণ পর্যন্ত exocrine gland বলছিলাম, তাই তো? এবার endocrine glands সম্বন্ধে একটু দুটো কথা, বেশি কথা নয়। There are two mode of functioning of the endocrine glands. We call them as paracrine and autocrine kind of glands. The endocrine gland can be divided into two parts. ধরো থাইরয়েড গ্ল্যান্ড বা এনি কোনো গ্ল্যান্ড, ইট ক্যান বি ক্লাসিফাইড ইনটু টু ডিফারেন্ট টাইপস। মানে এক্সোক্রাইনে এত ভাগ আছে, এন্ডোক্রাইনে অত ভাগ নেই। সো how this particular endocrine glands can be classified based on this modus operandi, how this particular endocrine glands or this particular element is doing or functioning or stimulating. Paracrine gland and autocrine gland, there are things which is known as, the paracrine means the neighbor, para means neighbor, মানে neighbor, আশেপাশে যে আছে, auto means self, right? Now if this glandular secretion is stimulating ourselves, then we call them as autocrine stimulation or autocrine kind of gland. If my secretion is stimulating the neighbor, that is known as paracrine gland. মানে আমি এমন কাজ করছি, প্রতিবেশী সবাই উদ্বুদ্ধ হয়ে বলছে হ্যাঁ আন্দোলন করতে হবে, so participating is coming. So paracrine gland of gland. Autocrine gland চলছে না চলবে না বলতে বলতে আমি এগিয়ে গেলাম, কেউ শুনলো না, আমিই করে গেলাম। কিরকম আন্দোলন হলো? অটোক্রাইন আন্দোলন হলো। বোঝা গেল?